16 PhD students, 8 awarded, 2 submitted, 6 working, and several postdoctoral fellows, master thesis, summer training. So it's regularly students comes, and sometimes we have to say no. And these are my research accomplishments: around 64 publications, 18 book chapters, and I have already means granted and applied for around 16 patents. International 3 US patents granted. One application is in the final stage. So like this, and I have developed around 8 technologies. Just in brief, I will give the description of these technologies in my later, but just I go through this. So I have developed a cyclic PCR, a handheld rapid PCR, one-step nucleic acid isolation using microfluidics, express cell-free protein expression system, magnetic nanoparticle-based one-step transformation of non-competent cell, dendritic silver structures-based SAS substrate, bioengineered mesoporous silicon nanoparticle-based dialysis system for kidney failure patients, and biomimetic antibody delivery system for antibiotic delivery system for treating antibiotic resistance. So some of the projects based on the sensor, I have given it summarized here. So biotic abiotic analysis, optical Raman spectroscopy based techniques. So where we use surface enhanced Raman spectroscopy, and it's in the current tier level is around 5 to 6, and electrochemical and impedance biosensors, DPVs, that we use, that is also in the range of 5 to 6. And handheld PCR, that is a specialized design that we have developed, that is as a current TRL 6 to 7, and we have just in the verge of commercialization. So the research goal of my lab or my research goal is to fabricate low-cost device, level-free sensor fabrications, optical and electronic, to increase the sensitivity and specificity of the existing system, automatize the system, and to miniaturize the high-end instruments for point-of-care diagnostics. So mostly you know that the problem is that we cannot take the samples to the lab every time, and it's better to bring the lab to the patient. And the concept comes point of care applications, and I am highly interested in developing point of care devices, handheld devices, benchtop devices, and with automation so that there is lesser complexity and anybody can use it. So I have collaborations with FZ Julie Germany, where I was there, and RVT Aachen, IIT Indore, Amity University, even NIRT Jabalpur with CDAC. So I have several to name. So just I skip this. So I come to this research and development, where we have shown that this SARS substrate. So Raman spectroscopy is very useful because it is direct sensing. You do not have to use any probe, and it gives you the chemical information. But the problem with the Raman spectroscopy is that the sensitivity is quite low. So it can sense up to millimolar level. So then that's why the use of this Raman spectroscopy was a little bit restricted. But with the invent of this nanotechnology, there is a concept called surface enhanced Raman spectroscopy, which has now evolved and take a boom, where different nanomaterials can be used as a such substrate. So there are two types of enhancement with the use of this nanotechnology. One is chemical enhancement, and another is electromagnetic enhancement. With the semiconductor non-plasmonic, basically anisotropic structure, we can do chemical enhancement, and we have shown that we can do chemical enhancement in the range of 10 to the power 4, 10 to the power 5. But with the plasmonic nanostructures, we can do the enhancement in the level of 10 to the power 9, 10 to the power 10. And that's why we have now started using this gold nanoflower. And this gold nanoflower fabricated by us, synthesized by us, can be used as a enhancement of this Raman spectroscopy. And we can detect up to nanomolar concentration. You can see the enhancement factor is 10 to the power 9, and we can detect up to picomolar concentration. So the problem with another problem with this Raman spectroscopy is that once you take it on a chip or a silicon wafer on a glass substrate, so the glass substrate gives very high background, so we prefer silicon wafers. But the silicon wafers, you have to find where there are the Raman substrate and where there are your Raman analytes. So what we have done a physical confinement in a microstructure, all the materials. So we have broadcasted our nanostructures inside this micro confinement, so that once there is a coffee stain, so what happens, you can see that goes to the periphery. So all the small analytes or small molecules, they always try to travel to the radial direction, outward radial direction. And now if you just squeeze this radial direction so that the center and radius merges, then you can get the signal from everywhere. So that was the idea, and it has been published. And once we do this squeezing of the structure in the micro scale, we can get this sensitivity very high, and we can get wherever you hit the with the laser, you get the signal. And that was the importance of this work, and it was published in 2018. So then we did tried with to increase the hotspots. So more the anisotropic structure, you increase the hotspot, more the junctions. If you see in this so structure, the hotspots and the dendritic structures have more hotspots, more junctions. And with this more junctions, can you see my pointer? So I don't know whether the pointer is visible or not. This is a pointer. So you can see this structures basically with the more and more junctions. What happens? The hotspots are increased, and once the hotspots are increased, the sensitivity, the enhancement factor also increased. So previously we have shown that the enhancement factor was 10 to the power minus 10 to the power 9. Now we have achieved the enhancement factor up to 10 to the power 12 and 10 to the power 15, and that's why we can detect, you see, femtomolar concentration of the, and in femtomolar concentration also you can see how the reproducibility it is. So everywhere where you hit, you can see detect the analyte. And with this sensitivity and this volume confinement, we can even detect single bacteria in 1 microliter solution. So that was a very good achievement. By this, we can detect up to femtomolar concentration of the analyte and single bacteria of, and this work was also published. So then we also work on, as I mentioned, that I worked on bioelectronics. So I fabricate my means electrodes by myself. So I have my own fabrication facility, and we can and you use this kind of biosensors where antibodies, aptamers, and they are used. And with this use of this different kind of biomolecules, we can immobilize them on these electrodes. And what we have found that if with this planar electrodes, if we want to increase the sensitivity, this gold nanostructures play is an important role. So the gold nanoflower and the gold nanoparticle was immobilized on this top of this planar electrode. With the increase of the surface area, you can see that the sensitivity of the gold nanoflower by using the gold nanoflower is more than the gold nanoparticles. So the gold nanoparticle increases the sensitivity, but the gold nanoflowers has more capability to increase the sensitivity due to the more surface area. And what we have done, we have immobilized different proteins on top. MAR is such a protein that is specific to bind to the mercury. It is from the MAR operon. So I'm not going to this biology in details, but the thing is that mercury can specifically bind to this MAR protein, and a signal is generated. And we wanted to measure this signal. So what we did, we purified the protein and we immobilized it on top of the electrode, and then we checked the sensitivity of the mercury. So with the DPV, you can see that it is very linearly giving you the data. And one more thing, just with this linearity, we can detect up to zero point 0.1 nanomolar. So around 100 picomolar detection we can do, which is quite fine with this detection of the mercury that is in the drinking water available in the different areas. So we also developed some sensors where we have used to detect the biomarkers. So for the biomarkers here, we have shown that how layer by layer deposition was done. So first it was salinized the ITO surfaces, then anti-PSA antibody was done, immobilized on top, and then this anti-PSA antibody plus prostate specific antigen binding was obtained, and with the blocking, and we can see that it can detect up to picomolar concentration of this. So you see, anti-PSA, 1 femtogram of PSA can be detected. 1 picogram of PSA can be detected with the linearity, with high linearity. So we can detect very low concentration, and that gives you the capability to for the early biomarker detection to predict the onset of the cancer. And this work is means under review, and also we have shown that different from patient samples, we can take out the data with the different dilutions. So with the dilutions, very low concentration of this patient serum sample, we went on dilutions, and at very low dilution also we can measure accurately the PSA level that was measured by other instruments, and it gives quite matches with the data that obtained from other instruments. So next we come that for different devices you or different sensors, you require one-step nucleic acid isolation, because nucleic acid is a key element. Once you saw during COVID-19 that nucleic acid is the best method to detect any pathogen, biopathogen. So not only the surface markers, the genetic markers are better than the surface markers or the protein markers. And for that reason, you require a nucleic acid isolation. But for nucleic acid isolation, there are several solutions and several steps. So what we did, we here devised a sandwich microfluidic device. In between, we have placed a aluminum foil, and this aluminum foil was anodized. And once this anodized, you get this kind of nanoporous membrane. And once this nanoporous membranes are prepared, we can directly just isolate the nucleic acid in one step. So you lyse the cells, and the nucleic acids will be staying on this, because you know putting a thread in the needle, it's very tough. So the long chain nucleic acid does not pass through the pore without any force. But the proteins which are the pores diameter is 60 nanometer, 50 nanometer, and the proteins which are in general in the of the range of 5 to 10 nanometers, they easily pass through these pores, and they go to the lower chamber and the west. And once all the proteins are passed to this west chamber, then you just move this with the pressure of the fluidic flow. Then it comes to this PCR device, and you can do the PCR. Here what we have done, we have also fabricated a on chip PCR. So this on chip PCR is a specialized PCR. So in general, the PCR takes place time for around 2 hours or 2.5 hours. But for this case, the PCR can be done in 20 minutes. So the main time required for the PCR is to change the temperature, not the PCR reaction. So here in this, we have designed three PCR temperature zones that can be heated, and the droplet with the assistance of magnetic nanoparticle can be moved on these three temperature zones, and the PCR can be done. So you see that these are the three temperature zones and this droplet. So the movie is somehow not working. So it will move on these three temperature zones, and this project was funded by BIRAC, and it was in collaboration with CDAC Kolkata. And we have designed more or less the prototype, the chip, and this magnetic nanoparticle can be moved on this with the use of magnets, and on top of the stepper motor, and we can program the stepper motor with the magnet so that it pulls this magnetic nanoparticle and the PCR mix on this, and the PCR is done, can be done in 18 to 20 minutes. So from this, we also developed a PCR, that is a handheld PCR, and the software. This handheld PCR the PCR can be done in just 15 minutes, and you can have multiple samples together. And the good thing is that you can operate all the chips at different temperatures, which is not available for any of the PCR device available in the market. So this is a handheld PCR device. So what happens that as this micro heaters, they don't require any time to change the temperature. They drastically drops down the temperature. So if a fidelity of the enzyme is 40 bases per second, then the 240 bases can be elongated in just 6 seconds. So you require 6 seconds for the elongation of 240 bases. In general, the other PCR will take around 1 minute to do this reaction, because the change in temperature for every cycle, and then 40 cycles, it increases the time with 40 minutes, and for each step 2 into 40 minutes, 80 minutes is increased due to this temperature change, so which is not required for our devices. So what we did also, In other work, we I just would like to show some of our other work that we did in our lab. So we have taken biomimetic platelets, the platelets from your body, and then we have incorporated the antibiotics in this platelet, and then we can kill the antibiotic resistant strains with this antibiotic loaded platelets. So this has been patented. The patent has been granted already, and we are in the final stage of doing the means final means animal model, and we want to go for commercialization. Recently we have received some fund for this one also, and you can see with this we can kill the resistant strains with the same antibiotic at very nanomolar concentration. So another work of our that group that we have done, that that patients has to go for dialysis every time if they have any kidney malfunctioning. So what we did, we have designed some silica bits which we made hollow from inside, and these are porous bits, and that can incorporate or take away the small molecules from it. You can see the small molecules, even dyes, iodine solutions can be taken. So it can take out the urea and creatinine from your body, and instead of this hemodialysis or peritoneal dialysis, you don't have to go anywhere for the. So our idea is that you take an injection just like insulin in diabetes. You take the injection of these particles, the dialysis will be done inside your body within the day, and you don't have to go anywhere. So this patent has also been granted. So, in general, the in the bacterial lab or molecular biology lab, those who work, they know that how tedious work is to do the transformation, because you have to prepare competent cells, store it, and once after 6 months you bring out the competent cell for transformation, you see that the competency has gone. What we did, we prepared some magnetic nanoparticles through which you can do the transformation without this competent cell preparation. So you don't need any means competent cell. You just take the growing bacterial cells from the secondary culture. You use our magnetic nanoparticle and plasmid DNA together. It will do the transformation, and it gives you the colonies just like and the thing is that we use instead of heat pulse, we used sonothermia. So we use ultrasonic ways to do the transformation. So that is the key thing. So another thing that people works that for the protein synthesis, those who work on protein based identification, antigen, antibody production, they have to go for mouse model, they have to go for bacterial model, mammalian cells. We have a cell-free system. You just require the plasmid to get the protein. So you do not require any other steps. And this cell-free system works like this. So you put the plasmid and that you give a sonication pulse. We have a cell-free system, and your protein is you get the protein in just 1 hour, and with very high quantity. So these are our business roadmaps that we are trying to final designing the prototype, including handheld fabrication devices that we and the other biotechnology products for commercialization, distribution of beta versions, and distribution of this many this self-free system. Also, we have got a BIRAC grant which is running. So we want to go for marketing at different technologies. As I mentioned that I have mentoring 2 startups. These products are with the startups, and we are trying to go for the market, and we are looking for business partners. So I am also open for any type of collaboration, including consultation. So our aim is to provide a cost-effective technologies, as I mentioned, not only for the healthcare, for any biotechnological centers, also industrial biotechnology also. So it fits with the National Health Mission and Made in India, and they are highly quantitative and competitive with the global market. So we have a functional nanobiotechnology lab, any kind of molecular biology, any kind of bacterial microbiology, and mammalian cell culture facility we have. So in our system, we have nano characterization facility in our lab, DLS, Zeta, TAM, SEM, AFM. I have a maskless optical lithography, plasma cleaner, electrochemical workstations, and others for characterizing and doing this electrochemistry and impedance spectroscopy. As I mentioned, all biological experiments. So we can fabricate electrodes, any immobilization of biomolecules, any requirement of help. I am there, I can help. Including whole cell, we have designed some whole cell sensors also which can detect the mercury. whole bacterial cells, whole mammalian cells we have immobilized on top, and that acts as a sensor. So we have also designed and synthesized aptamers which can detect different E. coli. That was the work that we did with CDAC Kolkata with the means call Grand Challenge call, and we can prepare any peptide or protein antigens using our cell-free system. So even they don't go to the inclusion bodies, and the yield is very high. So that's all from myself, and thank you. If you have any queries, you can email me and you can call me also.